11 and 12 of our notes. And yesterday we finished up by talking about proteins. Today we're going to talk about a special group of proteins known as enzymes. So enzymes can actually help to regulate conditions within a cell. And what they do is they convert your substrate, so what goes into a reaction, into a product. So they can actually put things together or break them apart. Enzymes have 4 unique conditions. The first is that they speed up chemical reactions, or the rate of those chemical reactions, by bringing those substrates together in the optimum orientation. So what that means is that when they're bringing together those substrates, those molecules that are being worked on, they will actually lower the amount of energy needed to get that reaction going. So they're going to lower your activation energy. Here on the graph you can see normally this reaction takes this much energy to produce the final product. But when an enzyme is being used, it's actually going to lower that amount of energy needed and still produce your same product. Because enzymes speed up chemical reactions, and because they're proteins, they're also known as organic catalysts. And a catalyst is just a molecule that speeds up a reaction. Remember to think that optimum is going to mean your best conditions, so it's going to create these good conditions so that reaction can occur. The second property is that enzymes are very specifically shaped. Remember we talked about proteins, and proteins are very specifically shaped. And so the same thing for our enzymes. Our enzymes are going to have a specific shape called its active site, where only very specific substrates can fit inside of it. So in this picture, if you look at part C, you have the same diagram in your notes. If you look at structure C, that's going to be your enzyme. And if you notice on your enzyme, you have this very specifically shaped area. That's going to be known as your active site. Your substrate, which is molecule A, is going to fit into your active site, and that's when it can be worked on by your enzyme. When it's complete, when the reaction has finished occurring, then your substrate is going to be released, and that's going to become your product. Okay? So the third property of enzymes is that they are unchanged during the reaction. So they are actually reusable. If you notice in this figure at the end, here is the enzyme working on your substrate. Here are the products being released, the end result of that reaction. And at the very, very end, your enzyme still maintains that same shape. So it can be used over and over and over again. Eventually, it can be worn down, and your body can break it down and use those subunits of the enzyme of the protein to make new proteins. But enzymes only work at enzymes work at their optimum rate in only some conditions. So they're only going to work at their best when they are in very specific pH, temperature, or salinity. And they can actually be denatured, or their shape can be changed, if there's some significant increase or decrease in one of those 3 factors. So here in this picture, they're showing you that normally this protein looks like this. Okay, this is its specific shape. But if we significantly increase something like the temperature, raise it up really, really high, then our protein can change its shape, or our enzyme can change its shape. Now it doesn't have that same active site, and because it doesn't have that same active site, it can't work on that substrate anymore. That substrate won't fit. Now the reaction rate of enzymes is highest in the optimum conditions. That means it's going to perform its job the quickest or at the best rate or at the best speed when it has those optimum conditions. So changes in the optimum conditions can cause the enzyme to denature, it can cause it to slow down, or in the worst case, it can actually stop working completely or actually cause death in your organism or cell. So organisms and their cells have mechanisms to help minimize those changes in temperature, pH, and salinity. In this figure, what they're showing you is here's the enzyme activity, how quickly or how well it's doing its job, how much product it's producing. And here you notice that for temperature, it performs at this temperature the best. This is going to be its optimum. Same thing for pH. And same thing, also increasing the amount of substrate, the amount of things it can work on, can also help to increase that activity. But to help prevent changes in temperature, pH, or salinity, you have some tools or some molecules such as buffers that can help maintain that pH within cells. It helps to regulate pH. pH is actually the acidity or it's how acidic or basic a molecule is or a solution is. And the value on this, it says that the value of 7 is neutral, below 7 are acids, above 7 are called bases. So pH, again, usually refers to how acidic something is. It actually stands for the potential of hydrogen. And 7 is kind of your key number here. Anything from 0 to 7 is going to be an acid. Anything from 7 to 14 is going to be a base. So you do have to remember those numbers. You have to remember that any number 0.3 or 4.5, any of those is going to be known as an acid on the pH scale, and anything from 7 to 14, like 11, is going to be a base on the pH scale. And then we just have buffers that can be used to regulate that pH and help to prevent the denaturation of our enzymes. Now in your summary box, I want you to go ahead and write 3 sentences to help summarize what are the key or most important factors or information about the enzymes that we've just talked about.